Okay, my fabulous friends, this is going to be fun. I have been working with a good friend of mine, Andy Moreno, who is a triple PhD bacterial engineer. He understands these things inside and out, and he is in business, and he supports me fully. Now, we're going to be talking about enzymes, which are things that bacteria create. He's the bacteria guy. Enzymes are created by bacteria. Enzymes come from ribosomes, and the bacteria create the ribosomes. We're going to get into this now, and it's going to be a little bit of a ramble because we just got off on everything after we went. It's going to be take a little while to go through this. But Andy is a fabulous mind, wide open to thinking, which is the problem with mainstream people. They have a set of rules that they go by in their head, like the speed of light can't be accelerated and all that business. It's just a lot of the things that everybody assumes and is such a core value, they cannot think outside of it. Andy is not that way. He's a thinker. He's a smart guy, and he's got the knowledge from the inside of mainstream, and the actual hands-on knowledge of working, because he has got a whole batch of labs. Last I knew he had like 19 of them that are in food service places where food is coming into the country, and he has to be sure nobody's going to get sick. That's what his business is. He owns a business, and they protect the food supply. So get ready. This is going to be really, really good. Okay, my friends, let me just set you up for this. I just got done with Andy. I want to preface a couple things. These are the bacteria we're going to be talking about, and they produce the ribosomes. Well, how come they're here and they're also here? Well, because these go floating around through the fluids in your body, and they end up just being absorbed by every cell. Every single cell in your body, even the blood cells, have a membrane around them, and they have ribosomes in them, and those ribosomes are chemistry sets. They are the tiniest things. There can be millions in every single cell, millions, and every one of them has a chemistry package built into it. And we're going to get deep into this. Andy is a top shelf guy, and he's a thinker. He's not just repeats what he was told in school, because he deals out on the street with this. He owns his own business, so he has to be sure he's right. He can't just take somebody's word for it, and he's been doing this many years, so he knows what to do, and he knows how bacteria respond and react and what they are and all the details about them. So thank God for Andy being able to spend a little time. All right, this might get a little geeky, but these are the ribosomes, those little balls that are inside the bacteria. When they come out, they create enzymes or proteins when they unsheath from this ball. But while they're in the ball, they can go anywhere in your body. Your body doesn't care. They're just like little inert things. They don't eat, they don't poop, they're not alive. They're encased in like a rubber ball, and they float through your body. And here's what happens is the bacteria pop them out. These are the ribosomes. They're so tiny. Just think about this. This is a single cell. Bacteria is a one cell, and there's thousands of them in here, and they squirt them out all the time. And those things just go free floating through the body, and then they end up, after they leave the bacteria, they end up collecting in the cytoplasm and on the endoplasmic reticulum, or cytoplasmic, I think it's endoplasmic reticulum. Yeah, endoplasmic reticulum. That's it. They glue on there because they are defenses. They're defensive little things. They're packages like bombs, and they float around through your blood. And when they're needed, they pop them open. Actually, they're in your blood, but they're also inside every single cell. There's up to 10 million of them, or millions of them inside a cell. It's absolutely incredible. So, and Andy did ask about the vagus nerve, and I said I really don't know that much about it, and nobody does, because all it is a gigantic conduit that carries all the signals from your body. Well, hold on. All right, all kinds of things go on inside your brain. There's a lot of activity. Let me show you how that works. This is a brain scan of a brain, and all of these are nerve fibers, and they run from like the medulla oblongata, and all the fibers run out of there. This is where the bundle of thinking part is, the neurons, but fibers have to carry them down and bring them everywhere. Now see this? This is a Mitchell Hedges skull. That's a real skull. That's a real crystal skull. You see those fibers in there? Exact same fibers. Identical. No difference whatsoever. The medulla oblongata, you see how it's forward and this one's back? It's because it was laying on its nose and its lips. And that's the only soft tissue that's preserved, is because it was in the mud with its nose and its lips. And all of the rest kept getting filtered and filtered and filtered and filtered and filtered with supersaturated silicon water that was pure water and pure supersaturated silicates. Somehow, I don't know how. But the nose was in the mud and the lips were in the mud. That's why it's pushed back and the brain is drifted down. But this is all these, every one of these nerves has to be insulated. That's what I was talking with Andy about, is the bacteria that live in your body and create the enzymes, which are the ribosomes. And they float around and they do, they may be doing a whole bunch of stuff outside of cells, but I believe mostly they go inside the cells and they accumulate in there, and whatever they're needed to do, they just get turned on by what's called a lysosome. That's like the director of all of the activity. It says, hey, we need some calcium over there. Hey, take care of that. Hey, oh, you over there, we need a little of this. We need some of this. We need some of that. Oh, there's a guy breaking in over here. Go and kill him. And these are the enzymes. But they're in these little capsules. They're not alive. They're just laying around waiting to be told what to do. And then you can't tell them what to do. Somehow they have to be moved and directed because they're not alive. So how do they get to where they're supposed to be? I do know they have a signal sensor mechanism on the outside of them. They're wrapped in some kind of a signal sensor. So it's like a ball like this. All right, well, actually it's very damn close. And it's a ribosome. It's a ball of chemistry. Now, it's got a couple little things maybe sticking off like that and one over here like this. All right, let's say that's the case. But basically it's the ribosome, it's the ball, but it's got a couple little fuzzy things sticking around somewhere. And when that fuzzy thing, with its charge, it has a certain charge, and it's exactly that certain charge. When it hits, click, boom, done. It opens up like this and it kills the bacteria or whatever it was invading you, or breaks down the carbohydrates and so forth, because it turns into one of these. From a simple little ball into one of these instantaneously when the lysosome tells it what to do. And when that happens, this can do more chemistry in 1 second than it could take up to 1,000,000 years to do without this making and breaking all these connections, these isotopes, which are half-life products. They're particles that are like atoms, but they don't have enough particles, or they have too many particles. And that's what enzymes work with. And that's how enzymes can make the heavy metals. They don't know how heavy metals are made. Ask a physicist, ask a metallurgist. They don't know. They say after iron, they must have been made in the Big Bang. No. The isotopes can jump them right up the line. I call it atomic bump. And when they decay, they don't decay downwards always. Sometimes they decay upwards. All right, so you could take gold and make it into mercury, but you could also make mercury into gold. That's what the whole thing about alchemy is working with the isotopes. That's what the alchemists could do. And they knew how to do it using enzymes, because that's the only way you can do it. You got to break off a bunch of little pieces and what's called nucleosynthesize. You got to take the nucleus and rebuild it. And that's what the enzymes do. They come in and go, and next thing you know, you got a big ball instead of a little ball. And they know how to do it. The bacteria know how to do it. The bacteria know how to program the enzymes, and then the enzymes, when they hit the thing they wanted to hit, and the only thing they're going to hit is what's on their surfaces, the program on their surfaces, to leave everybody alone until you see this guy that has one of these. If you see that, that's the guy you're going to attack. And then it does it. As soon as it hits him, all done. All right, so anyway, we're going to go through the stuff with Andy. He's a nice guy. And he's, like I say, the guy's a triple PhD. He's a very, very, very intelligent guy. And we talk frequently. And he's involved in this project. We're hoping to get a bacterial database over in Northern Ireland. Anyway, I am going to proceed. Okay, misunderstood microbes. Very misunderstood. Now they're saying recent scientific studies, very recent, have revealed the importance of microbes, and they're more and more important, as I have shown you or will. Despite common misconceptions, most microbes are more helpful than harmful to humans and the planet. They are the factories, they are the workers, they are the constructors, they are the sewage treatment system. They are the frontline troops. They program every single thing within your body, microbes. They are totally misunderstood because they literally control everything in your body, even the way you feel, your mood, how happy you are. It's all chemistry. And these are the producers of the chemistry as the microbes. It's called enzymes. All right, there can be up to 10 million ribosomes in one single cell. Every cell has them. And the reason they have so many is they can fight off almost anything instantaneously because they got all the troops all ready to go. All they got to do is be turned on. And the lysosome turns it on. Okay, my good friends, I have Andy Moreno with me today. And he is a specialist in bacteria. And what we're going to be talking about is bacteria. Now, that's Andy right up there. Andy, can you introduce yourself? Just give everybody a little idea of what you do. And by the way, he may not say it, but Andy's a triple PhD, and this is what he does is bacteria. So go ahead and do it, Andy.
My name's Andy. I work with a partner. We have a company called Andy Certified PCR Laboratories. We have many labs inside of food production facilities. Our main cause is that none of our client products will ever make anyone sick or kill them with pathogenic bacteria, primarily focusing on the three major categories of pathogenic bacteria in food, which is E. coli, O157A7, which is a subset of all E. coli, also known as the Big Six. Salmonella species, the entire species can make someone sick and kill them, and Listeria. We also focus on other bacteria and the sanitization of food prior to release to the public for human consumption. That is our mission. We've been doing it for quite a while. We use quantitative real-time polymer chain reaction detection methodologies, which is the detection of the DNA of these organisms as you can think of the internal workings that Roger's probably going to talk about. And those are then detected in an ATCG sequence. In other words, the genomic components are then compared against standard models, and that presence or the absence is then detected in a very rapid detection methodology to tell the food producer to ship the product to the public or not. Thank you, Roger.
Excellent, Andy, excellent. And basically, that's exactly what we're going to be talking about today. Exactly. I'm just going to start with the key component of life. And this just fell on me, Andy. I'm telling you, it all fit together just like boom. Ribosomes are little tiny balls that contain a big, huge program called an enzyme. Now, here's what it says right here. Ribosomes translate information. It's a program. A ribosome is a program. and it is encoded in a messenger RNA strand. Everybody's heard about that now. That's the thing that they use in vaccines. Now, they link together specific amino acids and polypeptides. I will show you all this in very great detail in a minute. They export these to the cytoplasm, which is a bunch of fluid inside the cells. As a matter of fact, right over here on the right is the cytoplasm is all inside the cell right here. Now, listen to this, and this just blew me away. A mammal cell like us may contain in one cell, Andy, as many as 1 million ribosomes. I had no idea. 1 million ribosomes. Each ribosome has only a temporary existence. So they come and they go. Now, ribosomes are free floaters. Another thing that I didn't know about. Ribosomes, here's what happens. You got bacteria, so you see in the bacteria. These, and this is this is Andy's area. He's a bacteria guy. How many species of bacteria are there, Andy? Have any idea?
Well, they're constantly being discovered and named. The issue is that we have a categorization system which puts them into general categories, but new ones, new variants, are constantly being mutated and discovered. Over.
All right. Do you know what that means? New variants. That means that they're evolving. That is evolution in real time. If it's a variant of the original one, its DNA is changing to create new ribosomes, because the ribosomes are the things that make you sick or they make you healthy. So let's just start with this ribosome. I'm going to assume that you don't even know about bacteria, and I'm going to talk to you like you're just a normal person and don't have any idea about this, and you just cut in on me when I'm wrong or whatever. Now, a bacteria is a single-celled organism, one tiny cell, very small, and this strand here, this pink strand, is the DNA, and it has a DNA just like we have. But it's very simple, and it says I am this kind of a bacteria species. I have this kind of a flagella, a little flipper there. My surface detects certain other bacterias and other things. In other words, it's like our skin and our touch. Only that, when something touches it, it's almost like in a chemical signature. And its program in life is to squirt out these little ribosomes, and that's all it's made to do. Now, that's one type of bacteria that squirts the ribosomes. Another bacteria will eat, like flesh-eating bacteria. They don't squirt ribosomes, they eat things, they break things down. But these ribosomes that we're going to be talking about right now are the mechanisms that protect you. So think about this. This bacteria is floating around through your body. They're just everywhere. And they're spitting out these balls. Now, what happens with those balls, they're right here. We'll call them triggers. And they're coming out, and they're so tiny that they can fit through the blood-brain barrier. And Andy knows, and a lot of people understand, the blood-brain barrier is there as a restriction, not to allow these huge molecules through there, because they can damage your brain. However, they will allow the ribosomes through. Now, the ribosomes, they're not alive. They're not alive. All they are is like a chemistry set. It's just like a little set of chemistry. It doesn't eat. It doesn't poop. It doesn't do anything. They're just floating around. But they do have a coating on the outside of them that makes them sensitive to something. And it makes them sensitive to a specific thing. So, in the case of a flu or a virus or whatever, if you have immunities built up, they will squirt out these little balls from a bacteria that is like one of your 105.279b bacteria or whatever it is, and it shoots out this exact ball, and that exact ball is made up of all kinds of little particles called enzymes. When it opens up, this is the enzyme right here. That's how elegant this is. And when I show you what it is, it's elegant. Now, these ribosomes, when they unsheath, they can do one of two things. So they can become an enzyme, which is a destroyer, or they can become a protein, which is a building block. The protein makes your muscles and your tendons and bone and all that stuff. And an enzyme comes out and breaks down your food, attacks invaders, and does all that sort of chemistry. And then there's other bacteria that run around eating up all the dead cells and doing all that stuff. So the bacteria really is 100% control of your body, 100%. Everything that's done requires a bacteria. And the reason is because they're enzymes. And enzymes are catalysts. And catalysts work, hold on, I got something in here that talks about catalysts. Well, let's look at this one. Never mind. I got to find the one about the catalyst. Well, maybe I don't have it here. But what a catalyst does is it breaks down other chemistry, and it breaks it down so fast. You know what? Let me find that article. Hold on a second, Andy. Okay, my friends, we're trying to figure this out and make this simple. I am going to make it simple. You are loaded with bacteria. There's more bacteria in you than there is you in you. More bacteria, and 99% of all the DNA programs that are in your body are in bacteria. And those programs create these ribosomes. Those ribosomes create molecules that are enzymes, and those enzymes work trillions of times a second to break down chemistry. So the chemistry we're talking about in your body is all produced by ribosomes. If you don't have the ribosome, you're not going to get the chemistry. No question whatsoever, you are not going to get the chemistry. If you don't have that bacteria, the ribosome will not be created. And what does the ribosome do? The ribosome does literally everything in your body. It breaks down your food. Here's the ribosomes, these little red jobbers. When they come out, they unsheath. When they do, they create the enzymes or proteins. Proteins build your body. Enzymes break everything, and that's what happens in your body. So between those two little devices there, coming out of bacteria, it literally controls almost your entire body. All right, it exists in the lymphatic system. That's all you have to know. It runs around through the lymphatic system, and the lymph flows through all this network of these vessels, and that's what brings these ribosomes around your body. These things are so small that just inside of a cell like this, and this is a single animal cell. You see it up here, animal cell? Every cell in your body has basically this much stuff in it. Now remember, I believe I mentioned that they can have millions of these ribosomes in a single cell, in one of these cells. Now over here, the ribosome is right here. There's a little ball that attaches on these endoplasmic reticulum, it's called. It surrounds the nucleus. But they float around in the cytoplasm, which is all the fluid. All throughout this whole cell, there's fluid. And it's saturated with these ribosomes. Now, every different type of bacteria, and as Andy said, there's thousands and thousands and thousands. I'm going to probably put the number, I would think there's got to be at least 50,000. And every one of them, almost, makes a ribosome, which is a chemistry set. So depending upon whatever happens inside that cell, you got the chemistry waiting right in the cell, just sitting there waiting to do the job to address that problem, whatever it might be, or to break down that food that's coming in, or to fight an invader that's trying to attack. But they just exist here because they're free floating in your body, and they seem to just get absorbed into the cell's inside the cell. And so they're just waiting there to be activated. And there is a little device in every cell. It's called a lysosome. And the lysosome is the one that sends out the chemical signal to say, all right, I want you to open up, and then it opens up. Otherwise, they just sit around. They don't eat anything. They don't poop. They don't do anything. They're just sitting around waiting to be activated. And if there's millions of them in there, you would suspect that certainly they're not all going to be the same, I wouldn't think. So you would have, all kinds of different chemistry capabilities. You wouldn't use them necessarily, but you're going to have them there. Now, the other thing is there's all kinds of different cells in your body. This one cell right here could be in your kidney, it could be in your heart, could be in your lungs, could be in your brain. It's still a cell. But it has a different job, and it sits inside of different chemistry. That's the other key. If you're in an acid situation, this particular cell happens to be somewhere down the stomach or in that, or if you're in some real mild environment, it's going to be different. So you're going to have all kinds of different chemistry. That's the key, it's the chemistry. And the only way we're going to know, and Andy's working on this with me right now, and Marguerite and a whole bunch of other people, is to try to get a database that says when you poop, in your poop is going to be all this kind of bacteria, because almost all your bacteria ends up coming out in your feces. And if we could just somehow have a standard database and accumulated data, all right, so you keep accumulating data, and you're going to find out that a lot of people that have, let's say, Parkinson's disease will be missing Lactobacillus reuteri, all right, and almost all the people that have some other disease are going to be missing Lactobacillus acidophilus or some other bacteria. And Andy knows there's a ton of species under, like acidophilus, Lactobacillus has, I don't know how, do you know how many species sit under Lactobacillus, Andy?
Well, Roger, as we've already discussed, there are general categories, but every day there are new designations of new variants of these categories of cells. And I'd like for everybody to remember, when we identify our names to these subsystems, they're numberless. And the better we get at detection and identification, the more we're going to be sub-identifying systems and organisms and sub-organisms. Over.
Yeah, right. I agree with you, Danny. And that's what I'm trying to put together now, is an understanding of how they do work together, because they absolutely do. You're right. This extremely sophisticated interplay. And the enzyme is this molecule right here. Remember I talked about, here, let's just take it for a start. The bacteria squirts out the ribosome. The ribosome is the ball. The ball goes scooting around through your body, and when somebody, which is the lysosome, tells it to do its job, it opens up, and then you have your enzyme. All right, so right there you've got the ribosome, you got the lysosome, and it also, I believe, when these ribosomes go to wherever they go, they claim that they are worked on basically wherever they end up going to, and changed. The ribosomes are changed a little bit, but I don't know if that's true or not, but let's assume that's true. When a ribosome opens up, they open up like this, and that is an extremely elegant chemistry package, and they do what's called click chemistry. So as that molecule, once it unfurls in the midst of a bunch of debris that it has to kill, it just keeps going around, click, 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 on each one of these little molecules that it bumps into, and they die instantly. It's like it's like electric shock, and I'm not kidding you. That's basically what it is. It's like just like an electric shock. So let's see, what do we have here? These things float around in the lymph and they float around in the blood. Now, if you don't...
Roger, can I interrupt here?
Yeah, absolutely.
Just a second.
Yeah.
You have mentioned in your personal research before that the human body has a subsystem that sends out these cells at rapid pace. Can you talk about the vagus system?
Well, I really don't know that much about it, to be perfectly honest, Andy, but I do know that there's something in you that senses an invasion. As a matter of fact, I'm going to show you the video of this, and we'll see it make more sense. Okay, here, I want you to see this. This is from some new photography techniques they use so they can watch the actual interaction of the lymph nodes, I'm sorry, of the white blood cells and the bacteria scooping up the ribosomes. That's my take on this. Now they think they're scooping up sugars. I say no, they're scooping up ribosomes. So here goes. This is Go Call, and it's on Seeker Channel, and he's with UC Berkeley. Here it goes. Now I turned off the sound. I've got it so that you can read it if you want to. So he's remembered, his hands stood up, his hair on his arms stood up. He was just freaked out. Now, this is a cell. All right, now I just stopped it. This big round little oval looking thing here is the is the white blood cell. And inside there is this guy right over here that's scooping up all of these little blue, which I think are the blue balls, which are the enzymes. And then he's going to go out into the fluid-filled highway, which has just been discovered. They didn't realize this. There's a fluid-filled highway that surrounds all of these white blood cell zones. And it appears to me that the bacteria has found a pretty good home in here. And the bacteria is squirting out all these little lymph nodes. All these little ribosomes. And then when they're required, somebody comes up and scoops them up. So I'm going to come back and show him scooping away here. All right, there's the white blood cell. Here he comes in, he's running. Did you see the little flagella? See that right there? That's the flagella. And he's scooting around here. And he's going to grab up a whole bunch of little blue ones, and finally he's going to zip right out into the fluid-filled highway. Now, that's the one that delivers the stuff. This one here doesn't deliver anything. It just looks, like he says, it's out looking around in the environment. And it reports back to this guy that grabs the things and delivers them. So that's the way it really works, is your ribosomes just sit around waiting to be scooped up and delivered wherever they have to go. It looks like to me, and I could be wrong about it, but what do you think, Andy? You think that might be right?
Well, I think what we see here is an actual video of, and I don't know if you remember this film, it was called Fantastic Voyage, and people had been miniaturized into a submarine. This was back in the 70s.
I remember, yeah.
And they went in to find a certain cancer, and they used lasers, and they were almost like the deep sea exploration. And your theory is that that same subsystem happens in our human bodies in very fast order. In fact, even Roger, you can talk about how fast the order is made internally, and then how the remedy, you call it the flagella-powered organism, goes out there and scoops up these invaders over.
Yeah, it's fast. It's as they said, and I think I showed you in the enzymes, when they do chemical breakdowns, which happen all the time. It happens just all the time. But when you have an enzyme, it speeds that reaction up, and it can speed it up millions, billions, even trillions of times. So they said, I was reading the other day that a certain sequence would have taken millions of years to happen, millions of years. It happened in less than a second. And what happens is they break up molecules like slice and dice and put them back together, and in half-lives. You've always heard of the half-life, and you always think of it as decay, of nuclear decay, a half-life. It only lasts in this particular condition for a nanosecond or 2 nanoseconds. Well, that's exactly what they do. They rip away some of the molecules or the particles away from the nucleus. It's called nucleosynthesis, where they take the nucleus and they resynthesize it into another nucleus. And they can go from one size smaller, and they can also go up. That's how heavy metals are made. Enzymes make them. We make them as living creatures, make the heavy metals. The astrophysicists think they had to be made in the Big Bang, because there's no other way for them to make these metals. It's impossible to make these metals. No, it's very easy to make them, but it takes a genius like an enzyme. It takes a bacteria.
Roger, could I ask you to explain very simply how we have, as our humans, we have no control over these subsystems that are doing it totally unilaterally, totally independent of anything we think or want to have happen? Their causation is totally independent. Can you address that issue?
Well, it is, but it's independent as long as you have the bacteria in you, which will make these substances, which are the enzymes, that work just incredibly fast. If you have the enzyme or the bacteria, you will have the enzymes, because the bacteria are the factories. They're factories that make the enzymes. So if you have that factory, you'll have the enzyme. If you have the enzyme, it will do its job, and they seem to do it flawlessly. Now, you do have genetic codes, the human genome, and it's in your DNA sequences. And a very, very tiny part of that DNA sequence is dedicated to the construction of your body, how many toes and fingers and eyeballs and all that stuff. And then there's a whole other chunk of that DNA that does all kinds of other things. And they control all kinds of processes in your body, but they can only control them if they have the raw materials. And that's why we are designed, when we're born, to be in the vaginal canal as much bacteria as you can get on you. Feces, it should go in your eyes, in your ears, in your nose, even, poop, everything. They keep the kids too clean. And this is all my opinion. I'm not saying to do this or don't do that, but you are a product of your interactions with invaders. And as long as your body withstands that invasion, it says, I know who that guy is. If he comes back again, send out 50279 to take him down. And that's what happens. You have a bacteria now that is in your body that is making these chemistry sets to deal with that invader. And as you get more and more invasions, you get more and more ribosomes. And that's why there's literally, like it said, millions in every single cell. So no matter what comes at that cell, you should have a guy in there that's ready to go kill it. And other cells are there, they have food in them and they had to be broken down, but it's the ribosomes. The key here is the bacteria. I can't say it enough. Bacteria, bacteria, bacteria. If we had a database that gave us a pretty good indication, like with this, we were talking about light spectroscopy, where all you did was take a smear of poop and you put it under these lighting conditions. And what it does is it gives off a little bit of a signature of frequency of what the frequencies of the things that are in that poop. And every single different bit of chemistry gives off a different signature of light. So when you take the picture of it, pop, just like that, you should be able to see all these different colors that relate to all these different bacteria. I don't care what you call them. But if everybody that has this pink bacteria is sick and has their left thumb fall off or whatever it is, you know that pink bacteria is going to be a problem. Now, then you got to figure out what that pink bacteria is. You understand what I'm saying, Andy? Right now, like you said, there's just zillions of these bacteria. Nobody's going to ever know them. We could name them all day long. But if we could tell if they're there or not just by taking a picture. And then eventually, go ahead.
Roger, could I break in at this point?
Yeah, absolutely.
And confirm two things. One, every human that consumes food will have either a positive or a negative response to that food based on their, we call it the microbiota, the organisms, numerous organisms that Roger's talking about living inside your digestive system. And what I deal with every day is E. coli, O15787, Salmonella, Listeria, organisms that in general harm everyone. What Roger is also focusing on, which is particularly interesting to me, is his discovery that in every human body there is a certain subset of standard bacteria which actually are very positive to help organisms survive, therefore the human survives. So Roger's theory, I am very intrigued by it, that every human can have a certain set of bacteria which are helpful to humans. And he also has a good thinking set of theories on probiotics. Roger, can you review that? If a person doesn't have the right set of positive bacteria, that can be supplemented. Roger?
Yeah, absolutely. The probacteria is what makes the enzymes. Now, they also make enzymes that you can take. But as long as your bacteria in your body is the standard bacteria, probiotic bacteria. Now, it's hard to reestablish that. Once you kill your bacteria in your body with antibiotics or something that kills a bunch of strains of bacteria, it's tough to get them back living in there. But I'm going to tell you right now, my theory is that literally 100% of invasive disease is caused by a problem with your bacteria. 100%. 100%. That's my theory. Now, why can I say 100%? Because every part of you is protected by a membrane. And the only way to get inside to the main source, your organ and so forth, is to break through that membrane. All right? The membrane houses the enzymes and the bacteria. That's where they live. That's the fluid-filled highway that protects you. So once your membrane goes bad, that's how the invasion happens. Here's cancer. All right, and cancer is pretty much, well, here it is right here. This just makes it simple to understand. This is a cancer growth. And there's your flesh up at the top, the little white and pinky looking things. And then you have this color layer down there at the bottom. This is the fluid-filled highway. This is a no-pass zone. You get through there, you're in trouble. It can everybody's got cuts and nicks and abrasions and all kinds of things going on up there. That's normal. But when that thing gets down and it breaks through this layer here and then it starts feeding on your really good tissue. This stuff up here, there's no real food to live on. Once it gets down in here and where all your good blood is and all that, then you get cancer. That's stage 4 cancer. In this layer right here, this little brownish looking layer, is where all your enzymes, you should be able to attack that and just kill that instantly. Now think about this. Just give this a little bit of thought. This particular invader is just a little tiny thing, little tiny thing invading you. In this layer is this fluid-filled highway. There's 4 times more fluid in that layer than there is blood in your body. 4 times. It's continuously flowing around your body. It's a fluid-filled network that does your whole body. And if it senses an invasion, it sends the troops. But you don't have the troops, you're in trouble. That's where the bacteria comes in. If you don't have the bacteria, you don't have the troops. The troops are the enzymes. So what happens? You get sick, and you can't stop it. And long COVID is, I believe, in my estimation, is related to this. All long diseases, all lingering diseases, all of the people that say they're easily infected, they're I forget what the term they use for.
The word is immunocompromised.
Yeah, there you go, immunocompromised. So if anything is out there that can damage you and you're near it, you're going to get it. All right, that's immunocompromised. Now, why would that happen? It's because of these layers of tissue here. There's what they call slurpees in there, small leucine-rich proteins. All these layers have to squirt around and slide and be pulled and pinched and twisted, and every layer, though, has to stay completely sealed. That's what a membrane does. It's a complete, unbroken seal. And if it breaks, whatever is, if there's some bad stuff, it is going to get through. Now, if there's no bad stuff, it can break. It's not a big deal. But if there's bad stuff and it breaks, then you're in trouble. And that's why you get so many in your lung infections, your nose, and all that stuff, because they're exposed to all of these different elements all the time. You're breathing them in and out. You got to have the right kind of mucus to protect you from these chunky little invaders. You want the oxygen, you want the exchange, but you don't want any. Okay, hold on to your hats. Andy mentioned this, so we might as well go down this road. Everything that I can find, literally everything, is made from biology. And the biology is so large that the average human mind is just, it cannot absorb it. These are comets and asteroids in space. All right, this is comet or asteroid Bennu right here. You see this? The NASA just went out and landed on this asteroid and picked up a sample and brought it back to Earth. It came back here last September. Before it came back, I said, I know what this is. It's a heart. And what they're going to come back with is blood. And I know precisely what blood is because they did a panel of all the elements in blood, and it's almost entirely made of metals. Now, look at the structure you have here. You see this right here, this over here? That's not a crater. That's a tube coming out. And there's another one over here. That's a tube. These are tubes from the heart. These are valves in the heart. This right here is the tendon material that's in the heart that holds on to the muscle. The all gooey stuff all left. Now, this is psyche. It's the same heart. Same type of thing, only this one must have swooped in and almost hit a planet or something, because it cooked and it smelted. Smelting means where you heat something up to over 2,700 degrees and it cooks off all the cheap stuff, all the lower end stuff, and you end up with metals. And blood is literally almost 100% metal. It's almost all iron, but then it has every other metal there is in it. That's why I know exactly what the chemistry is that they came back with. Plus, they came back with samples of it, and I can see they have phosphates, which is phosphates are the membranes, and they also came back with muscle sarcomeres. Did you see that picture, Andy, the muscle sarcomeres?
You and I reviewed that, that's correct.
Yeah.
So your theory, Roger, is that there are many levels of bacteria, gigantic planetary levels. There are also microscopic subsystems. Let's look at that heart on our scale versus the cosmic scale, Roger.
Well, that heart is big. Now, I'm not sure the size of that one, but let's take comet 67P. All right, here's 67P. What this is a hip joint. They had battles in space. They were blowing each other up like crazy. I mean, that was every word of this stuff is written. It's just that everybody always thought it's just too insane, and they still do. Even you show them the pictures, you show them everything, they still think it's insane. Now there's the hip joint. Now here's what happens. It broke right off of here. Where's my hip? Where did my hip go? Here it is. There it is right there. And it's broken off right there. Basically the same thing. Now there's some tendons, there's some muscles around here. We're right down to the bone. But here's what it looks like with the muscles and tendons and stuff. You see it? Now there's going to be some blood in here. And there's going to be some blood vessels, and there's going to be a main entry of blood. Let's see if we can find that. There it is right there. This goes back to 2015. I got a hold of NASA, and I told them, I said, this thing's biological. And of course, that didn't go all that well. Now, this is 67P again. You see that big blast coming out of there? That is barbecue smoke. Is exactly what it is. It's burning metals and blood. And the astronauts say that is exactly what space smells like. And they don't go out and take their helmets off. People say, oh, how do they do that? How do they do that? Well, they come in after the walk, and their suits smell like ribeye, or sirloin, whatever you want to call it. But they say it smells like steak in space. That's because everything out there is cooking. Now, when that artery facing straight out that direction, when it hits the sun just the right way, out blasts the blood. That cooks it. Now, over here, these little bitty ones, these are blood vessels. You see them going off in all the different directions. Every part of your body has to be fed with blood. So you have a main feeder, and then you have all the little blood vessels that feed the blood vessels. Feed the little tubes, the blood vessel tubes that lead up to the tissues and so forth. So that accounts for the artery, this accounts for the blood vessels, and this accounts for the steak smell of space. And look at this. This is the size of that big one. You see this right here? 40 meters is this wide. This is about 160 meters wide. That's about 500 feet wide is the artery. That's how big the artery is, 500 feet wide. You see these? They call these Dragon Balls. And they know something, because every single mission, like the one on Bennu is Osiris Rex, Apollo. They're all gods, and they're all something about the ancient past. They know something's going on, and they've thrown it in our face, and they think nobody's figured it out. Now, so here we got a heart. This is what a heart is. With all this stuff stripped off at the top. You see the tubes are stripped off, and you're down to where the valves are. And that's exactly what we have with Bennu and Psyche. See, this is all of them put together. Here's Bennu. That's the one they just took the sample from. Here's Psyche. You can't tell any difference. And here's the heart. You can't tell any difference from that either. Now, this is a heart with all the plumbing still attached. You see up here? We're down here in the valves. You see, we're here down here where the valves are for these here. All right, everybody with me now? We're down at the valves. What does that mean? It means from here up is all stripped off. So from here up is gone. So we have a valve here, we have a valve here. All right, and then that's this. That's this right here. And this one here didn't get cooked. If this got cooked up to 2,700 degrees, it'd look like this one. That's all metal. They saw this and they freaked out. They said, "Ooh, trillions of dollars." That's all they think about is money. And this is the same. They're the same thing. And I don't know if they're the same size or not. Maybe they are. Maybe they were from the same battle, from same guys. something happened out there in space, man. This is just, it's a nightmare, but you know, what are you going to do? It's there. Now, Umaru. Now, so far I showed you, we got body parts, we got hearts, we got hips, we got fingers. That's a finger. Now, I work with one of the top anatomists in the world, and I showed him this, and he said his exact words were, "That's the fingeriest finger I have ever seen." And he does not want to be identified. I said to him, I said, "Can I use your name?" And he said, "What do you want to do, ruin my life?" Now, this right here, I'm going to use my cursor here, from right up here, coming down, this is the tendon. Right here, you see all these little balls? That's where all the tendons invest at the end of the bone. The bone stops right here. So this is one big bone out to the end. This is the grip skin. It peels off because that's the heavy-duty skin on your finger. The other side of the bone is right here. That's the joint right there. And that comes running right down to center. And there's your vein, which is the black blood. This side is where your artery is. And down at the two bottom are the two tendons. Well, they're two tendons that come up over the top. And this is the bottom of the finger. And that's the joint. And there's no question it's a finger. Now, this is the grip skin up at the top here. You see how it's peeled back? Let me show you something. You know that I did all the CAT scans and all that business on my stuff. And now this was a giant finger that I have right here on my property. I'm going to show you the grip skin. This is the fingertip. I found this on my property. And I might as well tell you this. I think you know the story, Andy, about how I woke up one morning. I said, "There's something in the woods calling my name." You remember that story?
Yes, I do.
Yeah.
I want to make one statement before you explain that experience, Roger.
Yeah.
Your theoretical explorations have revolutionized human conception of our living universe and how we fit in an unimaginably complex system. Your observation and theory that we as human beings are living on top of the fossilized remains of gigantic creatures, and the chemical remains of these creatures are what we exist on, is astounding. This physical evidence that you found is unrefutable. Roger?
I believe, I agree with you 100%, but I have hit a stone wall, Andy, for 15 years, having the absolute, total, complete evidence. And right here, just anybody can see it. And the academic society they just can't handle this. But let me just show you this. Again, this all came off my property. And for some reason, like you say, this is a very complex set of situations we're under here. Now, I have to believe that I was contacted somewhere to say, go out in the woods and look. Because I woke up, I said to my wife, I just got a crazy feeling to go out in the woods and something calling my name. It's the exact words I said. I went out in the woods, and before long, I did find giant human being body parts. And this was one of them right here. Now, this is 3 feet long from the back pad here, which is where there's a little pad here between bones so that it doesn't scrub. So there's that pad, and here's the vein and artery. This right here literally is the fingernail. That's the fingernail of this fingertip. And this, all it is a tip. Now, I had to break this piece off over on the side. And the reason is you can't get anything out of any blood. And I wanted blood so I could take it and get a DNA test, which I did. And it's human. Now, if you look, most people can see the fingernail. And the thing is, I sold this property, so I don't have access to this anymore. But it's this is the fingernail that I found, and I broke the piece off right here. Now, this is the piece I broke off. Right there. This is the finger that prints. Just like the finger ridges on your, and these are sweat pores, these two little dots here. You see them up in here? Now, this popped off just like this. It wasn't even hardly attached. I just hit it with a hammer and it just popped off a big flat plate of it. So this is the grip skin. Now, remember Umaru. This is the grip skin here. Right at the tip of the finger. It's like we have a rubber glove on our hands. And that's what sheds off. So this peeled right off. Then underneath, inside here, it's saturated with blood. And it's not still red blood, but not too far off. Because there is a ton of blood in the human hands. You see this? It's saturated with blood. Once you get underneath that skin, you got all the blood you can ever use. So that's what I had tested. And now I had that one tested, and then I had another one. I might as well show you while we're here. The other one I had tested was this one. Here's the CAT scans. Well, here, let me just show you the fingertip. Or the hand. This is the hand. All right, that's a hand. That is actually a left hand. If you splay your left hand out, lay it flat back, tight, you have this tendon right here. And if you pinch your hand together, you have this little cleave right here. That's the pincher spot in between the big pad on the right, where it feeds off to your little finger. Your little finger runs off this way. The tendon runs right down the center. The pads are all around here. Here's the grip skin again. Remember the grip skin? On the fingers, the hands, the palms of your hands, your soles, that's the grip skin. And the thumb went off this way. Now, I do have some more parts to this. I have fingers and knuckles and all that. And I had this DNA tested as well, and this was also human. But mitochondrial D. The mother.
Roger, can I break? Roger, can I break here just for a minute and ask you to comment on the following? Every human watching your videos needs to stop considering rocks, hills, mountains as rocks. Rather, consider them and view them from fossilized remains of once living creatures. Can you make a comment on that statement?
Yeah, there's, like I say, as far as I can determine, there is nothing that exists that has not been alive. Even all the dirt and the eroded sand and all that stuff, that all comes from creatures' bodies that have morphed into stone. Even Jesus Christ said that the earth is a corpse and the earth is a body. And there's no way you can deny it. When you start looking at some of the landscape, well, even Ovid, who wrote long ago around the time of Julius Caesar, and he was an extreme historian, and his compilation was called Metamorphosis, and it said that the gods that were here, and they were here, could turn anything into anything. They could turn themselves into ants or mushrooms or grass or anything they wanted. And these gods were real, and the flood was real, and that's why all of my stuff, like the giants, like that's a product of the flood. Everything I have is completely, totally flat on one side, and exceptionally preserved. And as a matter of fact, I presented all my stuff to Yale a year before they wrote this paper. Exceptional preservation of soft-bodied creatures promoted by silica-rich oceans. That's when we had the global worldwide flood. And they're saying that my stuff, they say, was before any creatures existed. It was before there was any other life on Earth, which is totally wrong. They got this all wrong, but they use my research. Now here's what they say. This layer occurs worldwide. It's one layer worldwide. It's the Triassic layer. It's a red bed, gray clay, and black cap. And then they say, how do fossils remain so perfect? This is controversial. Well, I know exactly why. And it consists of exceptionally preserved soft-bodied creatures. Now, they think they were all before there was anything that had teeth or bones or anything. But I know this just totally wrong. I showed them all this stuff. I presented all this to Derek Briggs for years. And it ends up NASA paid them to do this because I sent it all to NASA too. And here's what they wrote in their paper on Yale. This is at Yale, Yale News. This is 2016. I submitted my paper on Academia EDU. It's called Fascia Facilitated Fossilization. That was on 2015. And I wrote the book about this 2015 too because I was so upset with Yale. And here it is right here. A Yale-led research team found this collection of marine fossils. Well, marine means they're underwater, and it's all around the world, the whole world, 100% of the world. There's nowhere that didn't have this flood. All right? And they're saying it happened hundreds of millions of years ago. No, totally wrong. They're saying this was from 540 to 580 million years ago. And it's found worldwide. Worldwide, that means there was a flood. Hold on one second, Andy.
Hold it.
All right, so they're talking about the Triassic era being where these creatures were, and they lived on the communities on the sea floor. That's the whole world was a sea. That's why. They say many of these creatures are outright bizarre and do not resemble anything alive today. Now they want to understand about the evolution of them and so forth, and are anything today similar. Now listen to this. It says a big part of this answer involves figuring out how these fossils were able to form. I know exactly how. It was a hot, saltwater, silica-rich flood, and it was because we almost got impacted by Venus. And when it did, it made the whole Earth flood, and it also brought up all the siliceous ooze, which is a very heavy ooze that sits on the ocean floor, and that's where the silicon came from. Now listen to this. They say these animals were entirely soft-bodied. They lived before the evolution of shells, teeth, bones, typically the only parts of the organism to become fossilized. No, they were giants, they were dragons, they had every single thing. Now, what the researchers realized was that the oceans during the time of this were much richer in dissolved silica than they are today. That's because it came up from the ocean floor. Look it up, siliceous ooze. And the silicon came up and supersaturated the oceans and filled in all the voids. That's why they say veins of quartz. All right, this is what they came up. Silica is quartz. They say veins of quartz. They are veins of quartz. Most of them are arteries. Some of them are veins. But the silica filled in those holes. It enabled the transformation of these animals to come into rock over a matter of hours or years. I told them it was very fast. I don't think it hours, but I made some. It didn't take all that long. But I didn't know about that they had to be in the boiling hot water. That was a new revelation to me when I finally figured that out. Now, so it says they could actually turn into stone within hours or years rather than thousands or millions of years. Now they're saying right here, the Idacarda organisms were rapidly buried by sand during underwater storm events. The whole world was underwater. and it wasn't sand, it was mud. They're trying to change it so that they could take credit for it, but they didn't. They just took my work. And it says, otherwise they were...
Roger, can I break in at this point?
Yeah.
This paper addresses the issue of metamorphosis, or the physical evidence of chemical transitions. This is something you're an expert in. From living to solid, and also from metal, human work to metal, to rock. You had a picture once of a World War II German tank that had sunk into a bog, and it is now transformed into a rock. Do you remember that picture?
Yeah, I'm not 100% sure of that one, Andy, but I can tell you what. Anything that is inside of a wet, moist, muddy environment, and there's transition metals, a lot of transition metals in there. They will work their way, especially into body tissues. Now that tank, I remember what you're talking about, and I can't vouch for that 100%, whether that turned into actual stone. It did look like it. However, I can absolutely 100% tell you that anything with body tissues in it, which always work with transition metals. Transition metals are the things that bring all of the products through your body. And they do it because they have a charge on them. And they have what they call ligands that attach around the metal core. And the metals in your body, you want to see what's in your body for metals? Let me show you something. I think it's this one right here. Nope, hold on, it's dipole flood theory. Let me get this out of the way.
Well, Roger, while you're looking at that, perhaps we could wrap up this conversation with the following observations on your part. And you don't need to bring up any evidence. You can completely deduce your theories with the following. A, you can answer these questions in any order you wish. Do we live in a living universe? And are these living creatures, their remnants, made up of these named chemical compositions? Because these are all names that we've given them. And do those affect how we live today?
Well, we are living on, we make things that are dead alive again. And Jesus said that too. He says, when you make what's dead alive, he's got a whole thing about it. But getting back to the menu and all of the stuff that's floating in space, they have that sample here on Earth right now. It came from a heart. This is whole blood. You see it, whole blood, analytical. This is what they're going to find in that. And these are the actual measurements, the concentrations. Now, it might be a little different because it's been exposed to a lot of ionization being out in the outer atmosphere, but it's not going to be too far different. All of these is what they're going to find in that sample. And they can do this with a click of a mouse. They can tell how, with spectroscopy, what is in those samples. Instead, they're talking about taking years and years to view these samples. And
yeah, that methodology, by the way, is known as mass spectrometry.
Yeah.
And it emits light through a magnetic field, and then the gravity can detect the specific metal or composition. So I'd like you to wrap up, Roger. I have really learned a lot. I hope that your audience considers the ideas that you have found on your own. You haven't been to college. You haven't confined yourselves to chemistry, biology, microbiology, astrobiology, or planetary science. You came up with all these ideas on your own. Over.
Well, yes, but that thing about not being at college, I've gone to colleges, lots of them. I went to college after I got out of the Army. I was very, very involved in the Army with nuclear stuff. I worked on nuclear, Nike Hercules missiles. And I got out, I went to a bunch of different colleges, and then I went through everyone just to confront them, basically. I realized that they were just totally wrong. Dipole electron flood theory, right here, this is the only thing that works. They have to change the nuclear model. It's not right. Dark matter is in the center of everything that you're looking at right now. Everything you're looking at has dark matter in it. Even light has dark matter in it. And it's in the center of all this. They don't know how gravity works. They don't know about dark matter. They don't know about light, the speed of light. They think the light never slows down, and these Webb telescope and all this stuff is seeing things 10 bazillion miles away. They could be right down the street. They have no idea where these things are, because all light comes through the, well, here, let me just show you something. Light comes through space, and when it comes through space, it has to come through all fields and all particles and all dust and all other light that's out there. And here they think it's not going to slow down. It is insanity, absolute insanity. This is what's in space. You see that? Light has to come through that. You think it just comes flying through there and doesn't have any interaction? They're absolutely insane to not understand. They have been destroyed by laws. Einstein's law of this and Planck's constant and all these different nonsense things. And all they do is make them up so that they can justify their wrong conclusions about the universe. The universe is not expanding the way they're talking about, pulling the light away, making it shift to red. It's just slowing down. That's all. And the cosmic microwave background has nothing to do, I don't know what they think it is, but all it is light that has hit so many little fields like this that it's not hardly moving anymore. It's just sitting there. Totally lost, absolutely lost. And
well, Roger, I want to close this conversation by concluding that your thoughts are totally off the books. And yet, you can prove every major point through your experiments, through your physical sampling, and your current methodologies. Now, we need more of your experiments with this light photon vortex, but the issue is for us, you have a lot to contribute, and we should not restrict you in any way. Over.
Yeah, well, I can tell you what, I have been restricted. They put me in a zone of people that are, they're not in the normal range of people. Let's put it that way. There are people that are so far off the scale, which I am, but they have no evidence. They just saying the Earth is flat and there's no space and there's all this crazy, insane stuff. And then they put me in that range because I am also saying this isn't right, this isn't right, this isn't right. But I have the evidence to prove it. I'm not just saying it. You know, I mean, and I got put into that box. I cannot get out. I cannot get out. So the normal.
I offer, Roger, that you not be concerned about what others think, but you continue on your open-ended explorations and contribute to the body of human knowledge without restriction. That's my advice to you. Over. Yeah,
well, that's what I am doing. That's all I can do, because everybody's, oh, you should try this and call this guy and do this and do that. I gave up, and nobody is interested, really. And the only ones that are interested, I feel they're only interested because God made them interested. I'm going to tell you right now, this is much more than just chemistry and physics and science and history. And this is eternity, my friend. And all of this stuff that they talked about is not just nonsense that they made up about giants and dragons to scare kids and make them go to bed or something. This is real. This actually happened. This is not nonsense. And I know you want to wrap it up, but let me just show you one thing right now. Google Earth. If I can get to Google Earth, just so that we can finalize this thing to show how ridiculously gigantic these creatures were, and that they were real, and they were talked about. And Typhon was talked about as a gigantic dragon And here he is right here. There's his head, and he runs all the way across North Africa. I'm going to show you this. And here he is attacking this gigantic fish. That is a gigantic fish. And here's his head and his throat. All right, this is the outside of his head. And here's his throat running down. And these are all dragon scales in this throat running down. You see the scales in here? All of these are dragon scales coating his throat, running all the way down here. All dragon scales. Now, on the other end, where his tail is, this just goes around through his whole body is here, and his tail is here, and there's still dragon scales way down here. You see the scales? This is a dragon. He was written about exactly for what he is. And he was attacking one of the gods who had turned themselves into animals to try to get away. And this one got killed. This was turned into a fish. It's exactly what's written in Apollodorus. It's word for word about Typhon. And his throat was cut right there. And there's the red blood running out in the desert from his cut throat. It's still red blood when it rains. And that's what makes all this stuff turn green. Blood makes things grow very, very green. And this is in the middle of the desert, is our dragon friend. And I want to tell you one thing. I know you want to wrap it up, but I just want to tell you this one thing. I was selected to make these messages. As I told you, it said go in the woods and look for something, and I found it. And then when I started searching, I got a message came up that said, these things are for you to see and for others as a test. And that blew my mind. It went away. I have no idea where it came from. And then several years later, when everybody, literally everyone, failed the test, my wife has still never even seen one of my videos. People don't want to look at this. My own wife has never seen one of my videos in 15 years. People just are dead set against this. But after 3 or 4 years, maybe 5 years, I'm sitting here looking at my screen thinking, I can't do this. God, you picked the wrong guy. I can't do this. And I was looking at Atlantis right here, because I think this is Atlantis. I'm absolutely certain it is. And I had been looking at this for a couple of years. As a matter of fact, I wrote a book. Well, I wrote a paper about it back in 11 of 2015, a book that says Atlantis and the Draining of the Sahara. So here I am looking at this, and I never saw the fish and the dragon for a couple of years, looking at the Sahara. Now, and I swear to God, this is an actual fact. I'm sitting here, and I don't remember what I was looking at, but I might have been looking at Atlantis. And I hear from behind, and I'm really literally in tears, saying, I can't do this. Nobody's going to listen. They're just not going to never going to happen, God. You picked the wrong guy. And I heard from behind me, look in Morocco. I swear, that's a fact. And I just did. I just jumped on and I ran right here and I looked in Morocco, and here's the dragon.
Roger, I want to add my experience to you as a person. You're from Digby, Nova Scotia. I served my missionary service there. Those people were from England, direct descendants of the area where Merlin the magician was an actual person. So I'm going to leave that for your audience to deduce. The people of Digby, Nova Scotia are amazing. They are theoretical, they are visionaries, and some people may have called them wizards in the past, but they were thinkers, deep thinkers, immersive thinkers. The sun that we revolve around can take up to 1.4 million sized Earths in volume. We do not understand scale. Roger attempts to introduce scale to us with this 1,200-mile long fossil. But we do not understand scale, even from the microscopic or submicroscopic atomic level of light. But Roger, in his brilliant mind, could take all those concepts and put it into an understandable format. Roger, I admire you and I praise you. And I have to leave at this time. Thank you so much.
Okay, Andy, I really appreciate your time taking, because I need somebody with some credibility to weigh in on this, and that I have not been afforded. All right, Andy, take care, my good
You don't
friend.
need anybody, Roger. You're a descendant of Merlin the Magician. An amazing group of people.
Well, I think I'm going to go make myself some amazing tea. Andy, you're a pleasure to talk to, my good friend, and thank you for the years we have spent together. All right?
It's excellent.
We'll talk later. Thanks,
Thank you
buddy.
very much.
All right, bye.
Bye now.